this presentation, we will review the structure and function of the three ancillary digestive organs, including a quick review of metabolism of the macronutrients. Content in this presentation is a compilation of thoughts from a variety of references and textbooks, not just from the class text. The three main ancillary digestive organs of the liver, gallbladder, and pancreas are illustrated here. The liver has a dual blood supply. About 20% is oxygen-rich blood from the hepatic artery from the aorta, and the rest is the nutrient-rich blood carried by the portal vein from the stomach, intestines, pancreas, and spleen. The liver is the most active processing center in the body. The gallbladder is a pear-shaped structure on the underneath side of the liver. Its main function is to concentrate, store, and excrete the bile that is produced by the liver. The pancreas is an elongated organ that has both exocrine and endocrine functions. Over the next 20 or so minutes, we are going to examine each of the organs in more detail, starting with the liver. The liver weighs between 1 and 1.5 kilograms, making it the second largest organ in the body. Approximately 1,500 milliliters of blood per minute circulates through the liver. Amazingly, the liver has the ability to regenerate itself, and our body can function with a liver that has only between a 10 and 20% functionality. 2/3 of the liver cells are the hepatocytes. These cells complete the majority of the metabolic functions of the liver. When nutrients enter the body from the GI tract, the liver receives them first, then it metabolizes, packages, stores, or ships them out for use by other tissues. The Kupffer cells are macrophages that are dedicated to the liver. They protect against the pathogens arising from the gut. The chelate cells are responsible for collagen formation and store about 90% of the body's vitamin A stores. The liver is often considered the nutritional gatekeeper of the body. It is the only organ capable of all of the major metabolic pathways of the macronutrients. The main functions of the liver are presented on this slide. We will chat about the first three in greater detail in just a minute. The final four functions of the liver include the storage and activation of the micronutrients, such as the fat-soluble vitamins, and the metabolism or inactivation of the steroids, such as aldosterone, estrogen, progesterone, and testosterone. The liver detoxifies toxic substances such as medications, alcohol, pollutants, and pesticides, and removes the byproducts for excretion from the body. And finally, the liver acts as a filter by removing bacteria and debris from the blood through the phagocytic actions of the Kupffer cells. Before we get into the specifics of macronutrient metabolism, let's take a moment to quickly review the larger picture of metabolism. Metabolism is the sum total of all of the chemical reactions that occur in living cells. The end products of metabolism are energy, water, and carbon dioxide. Cellular respiration includes all of the reactions that break down glucose, fatty acids, and amino acids to produce carbon dioxide, water, and energy in the form of ATP. Some of the metabolic reactions require oxygen. These are the aerobic reactions, while others occur in conditions without oxygen, or the anaerobic reactions. Anabolic reactions are those in which small molecules are put together to build larger ones. Anabolic reactions require energy as they are building a new or different substance. Examples of anabolic reactions include converting glucose into glycogen, linking amino acids together to make a protein, and esterifying fatty acids and glycerol to make a triglyceride. These are all examples of condensation reactions. Catabolism, or those catabolic reactions, are those in which larger molecules are broken down into smaller ones. Catabolic reactions release energy. Examples of catabolic reactions include hydrolysis reactions, or many of the digestive processes in the body. The four end products of catabolic reactions are glucose, fatty acids, glycerol, and individual amino acids. As the substance are built or broken down, they will use or release energy, many times in the form of ATP. Oftentimes, the hydrolysis of ATP occurs simultaneously with reactions that will use energy. This is known as a coupled reaction. Let's head now into the liver for a cellular respiration refresher. In the liver, galactose and fructose are first all converted into glucose. Then the glucose can be made available to the body's cells in several ways. When the body has sufficient glucose, the glucose could be used for immediate energy to fuel the body's cells. If the body has more glucose than what is needed for immediate use, then the glucose can be stored as glycogen in a metabolic process known as glycogenesis. Approximately 1/3 of the glycogen in the body is stored in the liver, and the other 2/3 is stored in the muscle. Humans have a limited storage capacity for glycogen. Additionally, when we have excess glucose, we can convert it into fat in the process of lipogenesis. As this takes much energy, it is not the preferred pathway. Typically, carbohydrate goes into carbohydrate storage and fat goes into fat storage. When there isn't quite enough glucose, the body can release the stored glucose from glycogen and/or produce new glucose from non-carbohydrate sources, such as lactic acid, glycogenic amino acids, and intermediates from the Krebs cycle. This is known as gluconeogenesis. In more dire cases, when there isn't enough glucose, ketone bodies, or ketones, can be produced from fat fragments and ketogenic amino acids. The ketones then can provide an alternate fuel source during starvation. However, when too many ketone bodies are produced, they can accumulate in the blood, leading to ketosis, which disrupts the acid-base balance in the body. In the cytosol of the cell, the process of glycolysis splits the 6-carbon glucose into 2 3-carbon pyruvate molecules. This step produces high-energy electrons and 2 molecules of ATP per each glucose molecule. Each pyruvate is then either broken down to produce more ATP or used to make glucose via gluconeogenesis. From here, pyruvate has a choice. It can enter into the anaerobic pathway or the aerobic pathway. The anaerobic pathway is chosen when oxygen is not needed for the activity. For example, when you need a quick burst of energy. In this pathway, known as the Cori cycle, the pyruvate accepts electrons to become lactate. This conversion frees the coenzymes so that glycolysis can continue. The liver enzymes convert lactate to glucose, but this reaction requires ATP. The glucose produced in the liver then heads back to the muscle for use. The aerobic pathway is chosen when oxygen is required and available, so for the more endurance type activities. Here, 1 carbon is removed from the pyruvate and released as carbon dioxide. This produces a 2-carbon molecule that combines with coenzyme A, which is made from the vitamin B pantothenic acid to form acetyl-CoA, which releases high-energy electrons. The citric acid cycle is a series of metabolic reactions that break down molecules of acetyl-CoA to carbon dioxide and hydrogen. It starts with oxaloacetate and ends with oxaloacetate being synthesized in the last step. Oxaloacetate is made primarily from pyruvate, but it can be made from protein, but not from fatty acids. Most ATP is produced in the final step of cellular respiration. This is known as the electron transport chain. Here, the energy from the high-energy electrons released in the previous steps pumps hydrogen ions across the inner mitochondrial membrane. As the hydrogen ions flow back, the energy is used to convert ADP into ATP. The electrons are combined with oxygen and hydrogen to form water. So what happens when carbohydrate is low or limited? So even when carbohydrate intake is low, blood glucose levels must be maintained in the normal range in order to fuel the brain. Low levels of glucose in the brain can cause confusion, seizures, and eventually coma. To maintain blood glucose within normal limits, when carbohydrate intake is low, glucose can be obtained from the breakdown of glycogen and from the synthesis of glucose by gluconeogenesis in the hepatocytes as well as in the kidney cells. Protein is broken down to supply blood glucose through gluconeogenesis. The glucogenic amino acids can be used to form pyruvate and oxaloacetate, and thus can be used to make glucose. Fatty acids and ketogenic amino acids cannot be used to make glucose because they are reduced to the two carbon molecules that form acetyl-CoA. Since adequate dietary carbohydrate eliminates the need to use amino acids to produce glucose, this is how carbohydrates are said to spare protein. To be metabolized via the citric acid cycle, acetyl-CoA must combine with oxaloacetate, which is made from carbohydrate. When carbohydrate is in short supply, oxaloacetate is conserved, so acetyl-CoA cannot enter the citric acid cycle. Instead, the liver converts it into ketones, which are released into the blood. Ketone production is a normal response to starvation or a diet that is very low in carbohydrate. Ketones can be used as an energy source by tissues, such as those in the heart, muscle, and kidneys. After about 3 days of fasting or low carbohydrate intake, even the brain can adapt and obtain about half of its energy from the ketones. The use of ketones for energy helps to spare the glucose and decreases the amount of protein that must be broken down to synthesize the glucose needed. Ketones not used for energy can be excreted by the kidneys in the urine. However, if ketone production is high or sustained for a longer period of time, the ketones can build up in the blood, causing ketosis. Symptoms of mild ketosis include reduced appetite, headaches, dry mouth, and a fruity breath from the acetones that are released. Severe ketosis is more often seen in those with untreated or poorly managed diabetes. Looking on now into protein metabolism in liver, the absorbed amino acids are deposited into the amino acid pool. The amino acid pool is all of the amino acids in the body tissues and fluids that are available for use by the body. The amino acid pool is comprised of the amino acids from the dietary proteins we consume, as well as from the breakdown of the body proteins. Protein metabolism in the liver includes the synthesis of non-protein nitrogen-containing molecules, such as the bases of RNA and DNA. Protein metabolism also includes the development of serum proteins, such as albumin and various globulins, and blood clotting factors such as prothrombin. The deactivation of the amino acids and the subsequent removal of nitrogen as ammonia is another function of the liver. Next, we have the conversion of excess amino acids into glycogen or triglycerides for storage. And then finally, we can synthesize other body proteins. Yielding energy from proteins is a multi-step process, starting with the removal of the nitrogen-containing amino group via the process of deamination. Deamination allows the remaining carbon compounds, either glucogenic or ketogenic amino acids, to be further metabolized into energy. The nitrogen-containing amino acid is converted to ammonia and then urea, and is excreted via the kidneys in the urine. In step 2, the deaminated glucogenic amino acids produce 3 carbon compounds that enter into the metabolic pathway as pyruvate. These amino acids can be used to yield glucose. In step number 3, the deaminated ketogenic amino acids produce 2 carbon compounds that enter into the metabolic pathway as acetyl-CoA. These amino acids cannot be used to yield glucose, but they can be used to yield fatty acids. In step 4, we see that some of the deaminated glucogenic amino acids can enter directly into the citric acid cycle to produce a quicker source of energy. Finally, in step 5, the high-energy electrons from the breakdown of the amino acids are transferred to the electron transport chain, where the energy is used to convert ADP into ATP. When energy is deficient, body proteins can be broken down into amino acids that can be used to generate energy or to synthesize glucose. This provides energy in times of need, but it can also rob the body of functional proteins. The most dispensable proteins are broken down first, conserving those proteins with more critical functions, such as those in the heart muscles. When protein intake exceeds the body's needs, and if the diet is adequate in energy, the amino acids are deaminated. The glucogenic amino acids are used to make ATP, while the ketogenic amino acids are converted to fatty acids and stored in the adipose tissue in the process of lipogenesis. In addition to carbohydrate and protein metabolism, the liver also plays a vital role in lipid metabolism. The hepatocytes are responsible for building and breaking down the various lipid components, such as triglycerides, phospholipids, and cholesterol, and then the liver repackages the lipids in various lipoprotein forms to carry materials to other body cells. The beta-oxidation of fatty lipids releases 2 carbon fragments that are used for energy when carbohydrate is limited, and the fatty acids can be used to make ketones. Finally, the liver manufactures bile that is necessary for the emulsification of lipids in the early stages of fat digestion. Triglycerides are metabolized into fatty acids and glycerol, with the fatty acids providing the bulk of the ATP produced. To begin lipid metabolism, the fatty acids are taken apart 2 carbon molecules at a time in a series of reactions known as fatty acid oxidation or beta-oxidation. The 2 carbon fragments combine with coenzyme A to form an abundance of acetyl-CoA. Acetyl-CoA enters the citric acid cycle to release energy or combine with other molecules of acetyl-CoA to make body fat. The 2 enzymes highlighted in green in the graphic are FAD and NAD, which are the coenzyme forms of the B vitamins riboflavin and niacin, respectively. In the second step of lipid metabolism, if oxygen and enough carbohydrate are available, The acetyl-CoA combines with oxaloacetate to enter the citric acid cycle, producing two molecules of carbon dioxide and releasing high-energy electrons that are shuttled into the electron transport chain. In step 3, or the final step of aerobic metabolism of the fatty acids, the energy from the high-energy electrons released from beta-oxidation and the citric acid cycle pump hydrogen ions across the inner mitochondrial membrane to convert ADP into ATP. In step 4, the glycerol is converted to pyruvate and enters the glycolysis pathway between glucose and pyruvate and can be converted to either glucose or pyruvate. Remember that fatty acids cannot be used to synthesize glucose, but the glycerol backbone can, although very inefficiently. This is an example of gluconeogenesis. Throughout the day, triglycerides are continuously stored and then broken down depending on the body's needs. When energy is ingested in excess of needs, excess energy can be stored as fat in the adipose tissue. Excess energy consumed as carbohydrate or protein must first go to the liver, where it is converted into fatty acids. These fatty acids are then assembled into triglycerides and transported to the adipose tissue in VLDL form. The enzyme lipoprotein lipase breaks down the triglycerides for both the chylomicrons from the GI tract as well as the VLDLs that originate from the liver, so that the fatty acids can enter into the cells for storage as triglycerides. When cells demand energy, hormone-sensitive lipase inside the hepatocytes stimulates the adipose tissue, responds by disabling stored triglycerides, and releases glycerol and fatty acids directly into the blood. If there is not enough carbohydrate to allow acetyl-CoA from fat breakdown to enter the citric acid cycle, then the acetyl-CoA will be used to make ketones. So essentially, excess carbohydrate and protein can be converted to fat, but they cannot be made from fat. Given the nearly 500 tasks that the liver completes, there are numerous laboratory tests that can be run to determine the functionality of the liver. Hepatic excretion tests, such as total serum bilirubin, indirect serum bilirubin, and direct serum bilirubin, measure the production and functionality of, you guessed it, bilirubin. When these are increased, it may indicate overproduction of bilirubin or impaired hepatic uptake. Bilirubin, remember, is the main bile pigment and is derived from the release of hemoglobin from red blood cell destruction. Serum alkaline phosphatase and Y-glutamyl transpeptidase, or GGT, are enzymes that might suggest cholestasis, which is the reduction or blockage of the flow of bile, and it can be indicative of liver disease. Elevated hepatic serum enzymes, such as alanine aminotransferase, or ALT, or aspartate aminotransferase, or AST, or serum lactic dehydrogenase, are indicative of liver cell damage. Serum proteins such as prothrombin, albumin, and globulins each measure specific proteins. As most blood clotting factors are synthesized in the liver, vitamin K deficiency and decreased synthesis of clotting factors all will increase the prothrombin and increase the risk of bleeding. Serum albumin is a measure of protein metabolism in the liver, and serum globulin increases with chronic liver disease. There are other markers of specific liver diseases as listed on this slide. For more specific information on these lab tests, you can pull out Krauss's Nutrition in the or Food in the Nutrition Care Process and refer to Table 29-1. The second digestive ancillary organ is the pancreas. The pancreas is about 6 inches long and sits across the back of the abdomen behind the stomach. It is a complex organ that has both exocrine and endocrine functions. The exocrine pancreas, mostly acinar cells and a few duct cells, comprise the majority of the pancreas. The acinar cells secrete the digestive enzymes that assist with digestion, while the duct cells secrete the sodium bicarbonate. The endocrine cells, made up of the island of Langerhans, release the hormones insulin, glucagon, and somatostatin for the absorption into the bloodstream. In order to provide a steady supply of glucose, remembering that the brain must have glucose at all times, the concentration of glucose in the blood is regulated by the liver and by the hormones secreted by the endocrine pancreas. Insulin lowers blood glucose by stimulating the storage of glucose as glycogen and the storage of fat in the process of lipogenesis, or through the use of carbon components to make amino acids. Glucagon raises the blood glucose level by stimulating the release of glucose from glycogen, stimulating the release of triglycerides from the adipocytes, or through the use of non-carbohydrate sources to make glucose. There are a variety of biochemical measures to determine pancreatic function. Secretin stimulation measures pancreatic secretion, particularly the sodium bicarbonate. A glucose tolerance test assesses the endocrine function of the pancreas by measuring insulin response to a glucose load. This test can be very helpful for those with diabetes. A 72 hours stool fat test assesses exocrine function of the pancreas by measuring fat absorption that reflects pancreatic lipase secretion. This test can be helpful with those with cystic fibrosis or steatorrhea. Fecal elastase is an enzyme most commonly used to determine pancreatic function. It is an indirect measure of the pancreatic exocrine function. The final digestive ancillary organ is the gallbladder. As mentioned earlier, the main function of the gallbladder is to concentrate, store, and excrete the bile, which is produced in the liver. The bile exits the liver via the common hepatic duct and is directed to the gallbladder via the cystic duct for concentration and storage. The cystic duct joins the common hepatic duct to form the common bile duct, and then this bile duct then joins the pancreatic duct, which carries the enzymes. Bile is composed of bile salts, fatty acids, cholesterol, phospholipids, bilirubin, proteins, and immunoglobulins that support the integrity of the intestinal mucosa. Bile salts are made by the liver cells from cholesterol and are essential for the digestion and absorption of lipids, as well as the fat-soluble vitamins. In conclusion, the three ancillary digestive organs of the liver, pancreas, and gallbladder function together to help achieve proper digestion, absorption, and metabolism of the nutrients we consume. As an example of the synergy of these organs with the GI tract, let's think about when the duodenum senses the presence of chyme. When this happens, the hormones cholecystokinin and secretin are released. This, in turn, stimulates the gallbladder and the pancreas and causes the sphincter of Oddi to relax, allowing the pancreatic juice and bile to flow into the duodenum at the ampulla of Vater. All this so we can enjoy that luscious, rich piece of dark chocolate.